Good afternoon, everyone, and welcome to the fifth edition of WWGM's annual conference. It's a very special year because it's the fifth year that we're doing this. My name is Alok Bhattacharya, and I'm a trustee of WWGM and a person who lives with GNE myopathy myself. So today we have an exciting set of talks where you will hear from a number of leading scientists who are working in the field of rare diseases as well as in GNE myopathy. And some of them are working towards finding a treatment for us who are living with myopathy. At 5:30, we're also going to have an interactive panel with doctors where they will answer questions from people living with GNE myopathy. And finally, we will have scientific discussions for people who speak Hindi. So it's going to be the last session of the day. So for those of you who want to understand in more simple terms the scientific discussions that we've today, do stay for that. So we inaugurate the conference this year with two eminent scientists. Dr. Alok Bhattacharya is the managing trustee of World Without GNE Myopathy and professor of biology at Ashoka University. He will be speaking on updates on the activities of WWGM. Next, Dr. Rakesh Mishra is the director of the Tata Institute for Genomics and Society. He will be providing an overview of the work that the Tata Institute for Genetics and Society is doing. And they're doing a lot of work on rare diseases. And we're hoping that they will also include GNE myopathy in the programs that they're doing. After each of the talks, we will have a short time for question and answers. So please do reserve your question and answers for the end of the talks. With that, I hand it over to Dr. Bhattacharya.
So, thank you, Shilpi, very much, and thank all of the speakers who have actually, they've given their valuable time to be with us today. And also like to thank the participants who are here to listen to some of the development that is happening. So I'm just going to briefly tell you about what WWGM has been doing for last 1 year or so. This is an organization working to find cure for GNE myopathy and other rare genetic disorders. So basically it was set up in June 2015 as a patient support group and advocacy group. And our mission was to generate awareness about GNE myopathy among patients. I must add that before that, hardly, people knew about this disease, even among the clinical community. Only few clinicians, and some of them would be here today or already here. They're the ones that they were actually working on GNE myopathy. So one thing has happened in last 7 years is that a lot more people know about this disease, not only clinicians, common people, as well as scientists. And our major focus is to accelerate development of a therapy for GNE myopathy, and because there's no therapy available as of now. And of course, make this therapy available and accessible to all patients. In fact, we are fighting at various fronts to make all rare disease therapy available to common people. And for that, we believe that it should be made in India. And to provide support to patients for leading a fulfilling and safe life. We run a WhatsApp group. Shilpi does that and is very successful in binding the patient community together and also to get together. So, and how do we do it? We have a website, and our website has been revamped now. The website gives all the information about it, the disease, the diagnosis, the research efforts, activities, policy, etc. Simple GNE-myopathy.org. The other thing that we do, like this conference every year, we have been organizing this meeting.
Can't hear you.
Is it coming broken or it's not coming at all? Okay.
You're not audible. I don't know. Can other people hear?
I also can't hear.
You're not
Okay.
audible. Yeah. Can you please?
Okay, let me see. Just let me.
Very sorry about that, everyone. Some technical difficulties.
Let's see, can you hear me now?
Yes.
Yeah.
Okay. So I talked about, could you hear about, we have made a new website, revamped the website so that the information would be available much more easily to people. And like this year's patient day, we organize patient day every year. And these patient days have been quite popular. And of course, we put up the video link and those are also watched by various patients. Like this one, last year's patient day, which was quite popular and a lot of people participated in it. We are also participating in various events. And for example, we participated in Indian Society of Clinical Research and how clinical research is enabling transforming life of patients. Uh, uh, for example, various issues about regulatory pathways, operation challenges, patient perspective. I am also a member of the ICMR committee, which is a consortium for developing R&D and therapeutics for rare diseases. And we participate in various patient advocacy group, one called by the Ministry of Health. So essentially what we are doing is that we are participating or we are being invited to participate in various meetings to develop various issues about it. Is it anything to do with therapeutics, policy, etc.? We have a fairly strong voice in this community. Um, Rare Disease Day is the last day of February, 28th February every year. And this year's Rare Disease Day, WWGM conduct a month-long social media campaign, post and videos on Instagram, Twitter, Facebook to raise awareness of it. In addition, Shilpi wrote, in Rare Disease, wrote a personal essay to raise awareness about it. And I and Sudha actually co-authored a piece in Express Healthcare about rare diseases. So we have been involved in a lot of these media campaigns, policy issues with government and others. So we also have been trying to get our scientists to write grants for getting money to do rare disease research. Dr. Munia Ganguly from CSIR, IGIB, and us, we wrote a grant from ICMR and we got funding for a non-viral vector development and assessment for GNE myopathy cases. And Dr. Ganguly will be actually talking a little bit about what she has done. So fairly recently, last February, we got the funding for this. We have supported very small way based on the funding that we received, research projects. One of them, a major one that we are trying to develop is a model system to which can be used for testing GNE myopathy drugs. as well as, which will help us to understand what is the problem of it. It's been given to Dr. Samba Sivan from Indian Institute of Science Education Research, Tirupati. We also been working at various capacity to push the frontiers. And one of the problems that we are trying to tackle is that why GNE myopathy affects skeletal muscles. The gene is there in every cell type, but why it affects only skeletal muscles. So one of the development that came out, one of the outcomes that came out from this study is that there are different isoforms of GNE, and only a specific isoform is expressed in muscle, which is different from other tissues, which could actually explain in some way why skeletal muscles may be affected. What is a new activity? And this has been, we have been occupied with this for last year and a half, trying to develop a mega scale project called MyoMission. And this is a major mission mode research proposal for finding treatment for neuromuscular genetic disorder. We are targeting 3 diseases: GNE myopathy, limb-girdle muscular dystrophy, and Duchenne muscular dystrophy. This proposal has been submitted and went through 2 rounds of review. One round of review, then there was a presentation that we made, and after that we have modified it, and the revised version has been submitted a few days back, and we are hoping that we'll get some money out of it. And this is the proposal, actually, my mission, National Mission on Neglected Neuromuscular Genetic Disorders, and it was submitted to Department of Biotechnology. And what is this MyoMission all about? It actually takes care of all the various component of drug discovery. So you have, starting from understanding biology of diseased muscles. That is very important to understand what happens in the muscles and how we can actually tackle it. One of the recent studies that just came out from USA actually shows that there is a defect in a pathway called autophagy, and there are drugs available that can treat it. And if you make a drug, you need to test this drug somewhere. So you need animal and cellular models. You need outreach program to link patients, clinicians, and get them to participate in drug development process. There are clinical centers that involve in diagnosis, management, and natural history. Natural history is very important because how the disease progresses over a period of time is necessary, because that helps you to design, let's say, clinical trials where there is no placebo control needed. Then you need data center and computation, where data repository is important for this thing. You can develop disease progression models, mathematical model, disease prognostic markers, etc. That is something that that is very important, and you probably hear from Dr. Mishra something about that. And of course, you drug and therapy development, you need to develop ways by which you can test whether your molecules are working or not, and other modes of this thing. So this is essentially what the MyoMission is all about. It's all about developing therapy for 3 different diseases. So the broad aims are enhancing capability for diagnosis, clinical management. Having a patient cohorts is very important. That is a group of patients who can be used for testing your drugs. Okay, data center where all the data will be there, analyzed to check, for example, your drugs are working or not, etc. You do need a place to do that. You need centers which can have those, so that eventually future clinical trials can be done. And then, of course, development drugs and therapies. We are trying multiple, and that has been one major problem with the agency. They say, why you want to do so many of them, so many different types of therapies? And we say that we don't know which one is going to work finally. And we need to develop all the technologies in India, so that some technology may work for one disease, some technology may work for other diseases. And then we need capability for manufacturing some of the high cost for carrying out not only clinical trials and eventually this thing. What we are proposing in this is to become Atmanirbhar on the drugs. We should not be depending on Pfizer to give us a multi-million-dollar drug. None of us can afford that. There are a whole lot of 18 institutes are involved, and there are 38 investigators that are there. That's a fairly large community, and these 38 investigators come from 18 institutes. And at the time, TICS was not part of it. I think future TICS will be part of all our development work, and still TICS can actually participate in this project. And you can see that they're mostly National Institute of repute, starting from AIIMS, Apollo, Ashoka University, BITS Pilani, Bombay Hospital, Indian Institute of Science, ICERS, etc., etc. So all big institutes around the country are involved in this process. So we have started a new activity, and it's part of our blog, part of our website, a blog that would be talking about rare diseases. And we are hoping that you will see these blogs every now and then. The first blog came out about how parent groups actually help in development of the drug. It's about phenylketonuria. And already more than 1,200 reads have been seen in this, and we are getting a lot of very good comments about it, and people want more and more such blogs that is coming out. Okay, so our WWGM based on funding by the trustees, funding by the others, and it works in a shoestring budget. Everything is done by us, the trustees, and some patients do help us in running the show. So we obviously need money from everyone to run this thing. And we need to raise the level of funding. If we can raise the level of funding, our activities will also go up in a way. And this is kind of an appeal to everybody. So we have an upcoming event coming up, and a lecture by Professor Jason Doles on 1st November. This is hopefully you'll be able to listen to this. And this is the work that I was talking about. They have actually done very interesting recent work on it, and he's very interested to make a presentation, maybe eventually collaborate with us, collaborators in future. We thank you for your support, and I'll be happy to answer any question.
Thank you, Dr. Bhattacharya. And if anyone has questions for Dr. Bhattacharya, please do raise your hand or put it in the chat box. We have a few minutes for questions. So if you have any questions. And in the meantime, I would like to reiterate the appeal for funding. As you know, we are all donating our time and efforts to WWGM, and the funding is really important for us to be able to extend our work in different directions. So please do contribute to our work in whatever way possible. Are there any questions? Any questions on the work that we are doing? Okay, so I don't see any questions. Maybe they'll come at the end. I don't know. So with that, maybe then I would invite Dr. Mishra to deliver his talk. Dr. Mishra.
Yes, I'm trying to share. Okay.
Yes. Maybe you can make it full screen.
Yeah, that's what I'm trying. Yes. Can you see full screen now?
Perfect. Thank you.
Great. Great. So thank you, Shilpi, for organizing this, and thank you all, especially Alok, for inviting me. I mean, he has been really an inspiration for many things for many of us who developed a career in science in past few decades, and it's been so nice to talk, interact, and help us in various occasions. And it's really my pleasure created with this activity. So I'm going to in the context of WWGM annual conference, I'm going to tell what we are doing in TICS. So I will maybe take a few minutes to just tell about our general activity, I mean, a few other activities, and then I'll focus more on what we are doing in in respect of rare genetic disorder. One disclaimer that probably I am the least qualified to speak on this topic, but I had an institute where we have now a major program, and I was very keen that in TICS we start rare genetic disorders, which is lots of things can be done. I think there is a less attention from institutions in a really serious long-term mission mode to solve the problem. So it was a few months back that we started, and maybe now close to 1 year ago. And I think 3-4 months back we met Alok. I met in Delhi in some other occasions, and it was really very nice. Since then we picked up together, and I'm sure very soon we are going to do something really in the labs, which I'm really excited. So I may be ignorant about the details of the disease, but I really share the determination to do whatever is possible today for these diseases. So just few words about Tata Institute for Genetics and Society. This is an institute funded by the Tata Trust to solve societal problems in healthcare and agriculture using advances in science of genetics and genomics. So we use cutting-edge science and technology to solve societal problem. All our programs are focused on societal relevance. We just don't deflect attention into basic blue sky research. For there are other institutions. Our purpose is to pick the problem and do the brainstorming how to solve it, and then go all out in terms of collaboration, outsourcing, developing platform techniques, and solve it. And this is a program driven organization. This is not the PI driven organization with stakeholders on the board from the start. So that's our very clear cut mandate which we have set for ourselves. We are in a Bangalore Life Science Cluster, which is in the vast campus of University of Agricultural Sciences, and we have 4 institutions as a part of this cluster. NCBS is one of the premier institutions doing fundamental research. Then INSTEM is the DBT Institute. NCBS is a part of PIFR, which is the Department of Atomic Energy institution. Then we have INSTEM, which is a DBT Institute for regenerative and stem cell therapy. And then we have CCAMP, which is a kind of incubator for companies for biotechnology products and services and technologies. And of course, TICS, which is a Tata Trust funded institution. So we have 3 programs. One is infectious disease, rare genetic disorder, and crop improvement. These are the 3 programs that we have initiated. And our model is to collaborate, because we don't want to spend years in building big infrastructure and then getting tired in maintaining them. Instead, we collaborate with institutions. So we are located actually in INSTEM building. We have 2 floors of the INSTEM building in one wing, and that's where we operate. And we, as you will see towards the end, we really believe in collaborative models where we have some core technology platform, but the rest of the thing we do with our collaborators. So TICS collaborates with research institutions, with the industry partners, NGOs and trust, patient groups, farmer groups, and then clinicians, hospitals, and government agencies, because it is very obvious that if you are really trying to find a solution, you need to keep all these components on board. Otherwise, the final product or finding will not reach the real end user. So, as I said, I will just take a couple of minutes to tell you what we are doing in two other programs, then go to rheumatoid disorders. So, in infectious disease, we are doing environmental surveillance and disease ecology, and then antimicrobial resistance, which is a big problem. We are gearing up to start activities in that concern, and then vector control, where we want to find innovative novel methods which are environment friendly to control mosquitoes and hopefully other disease vectors. And I'll show you what we are doing for disease surveillance. So we are doing the surveillance, sewage surveillance in Bangalore, all 28 sewage treatment plants. We get samples every week, and then we assess the COVID load, and you can see this pattern here, how we monitor, and every week, we give the data to BBMP, and then this data is why I'm showing in Bangalore average. In fact, we get the data in ward wise, because 28 STPs, and then we tell the BBMP, that is Bangalore Municipal Corporation, where disease is increasing or decreasing and so on, and they take necessary action. So we also do surveillance. We have a very close collaboration with CCMB in Hyderabad, and we do environmental surveillance here, not in STPs, but in open drainage, 20 of them. And again, we do the same thing and generate information. So this is like in different collection points, you can see the disease going up and down. And we, both in Bangalore and Hyderabad, we do genome sequencing of these sewage samples and get the idea about what variant is going around and if new variants are emerging and so on. As you can see, Omicron, this is what from the third wave, and it came, and this is another way of showing the variant situation. So that is the, and then now we are, this program, surveillance program, is funded also by Rockefeller Foundation, which is very generous funding of $9.5 million. 2 more years of that are to go, which I'm collaborating, and where the Bangalore, Hyderabad, Delhi, and Pune are the clusters where we are developing this surveillance, not only for COVID, but for other infectious diseases, and plan is to hand it over to the municipalities or small civic bodies in various places. Then about vector control, we are looking at various aspects of intervention. So for instance, replace the vegetation that sustain mosquitoes, and the way mosquitoes interact with us, what can be repellent, attractant, and what are the other methods of where study the behavior of the mosquitoes and their mating behavior and disturb this process to control them. We have set up now olfactometers to assess, quantify attractants, repellents, and design little ecosystems to control the vectors. And I'll just few seconds about our next program, which is crop improvement, where we are doing 3 things. So one is mutation breeding, which means generate mutations and screen for the characteristics, and then breed them back into the variety that you want to bring in those traits. And the genome editing, because now genome editing is acceptable to introduce new traits. And pest management, one day can we have a pesticide-free crop system. And genome editing we are doing for the bacterial blight resistant rice, which already we have made. Now we have to cross out the vectors and all, and this will be ready to go maybe next year. And fungal blast, similarly, brown plant hopper, we have made the line which is resistant to it, and we are making herbicide tolerance lines so that the rice can be grown with less water, because currently it's not actually tenable to use this much fresh water for growing rice. Now, then with that, I come to our main discussion today, which is rare genetic disorders. There, we are handling multiple ways this problem, disease modeling in the lab conditions and novel interventions. Another aspect is diagnostics and screening. Maybe have diagnostic which can be large scale, and therapeutics. And I really don't have to tell this audience that what is the burden of this, although we call it rare genetic disorder, but they are really not rare in our country. And as Alok just mentioned, the intervention that are, first of all, they are not available for most of things. If they are available, they are just prohibitive for most of us. So with that, I think although it's a very challenging problem, but I think and that is our team. So you can see we have a strong team now of 8 people, and I am the kind of coordinator of this team, and we are really going full force for the trying to do something impactful for rare genetic disorder. The reason is that now in recent 10 years or so, the technology has really taken a really very high level of genomics, also genome editing, and then many other new therapeutics approaches, which some of them I'll touch upon. They really create now possibility of offering intervention which was not possible some time back. But if we are to do that, then we really have to work together, because we require to first of all learn the problem and find out the ways of solution solving it, and then a partner who take the solution to the end users, and then interact with society, very important, and with the policymakers, so that solutions are acceptable, and government supports this, because this requires a lot of investment as well. So with this team, I am just going to just really give a very overview of the kinds of things we are doing. As you will see, there are, I mean, you can really talk from starting from epidemiology all the way up to public awareness. Many steps are there, and our aim is to intervene one or more of these steps that can significantly add value towards diagnosis or treatment. So that's our effort, and we are in a one of top stem cell institute in the country. We share facilities with them, so we are doing several things. One is, for example, cell or gene therapy platform, which is being set up, where you can make take the cells out, and we have indigenized most of the growth factors so that it can be 80% of the cost is removed. We can grow them in the lab, which we will hand over to some industry partners who can then supply at very low cost. And then we are also trying alternate CRISPR-Cas systems so that we don't have infringe anything, and repair the cells, try them, and then they can go to patients. And in parallel, we are also making pluripotent stem cell based platforms where you can try many new drugs, or repurposing drugs, or trying new ways of enzyme replacement therapy or mRNA therapy. And I will just give some examples of what we are doing, what my colleagues have shared the slide with me. So all of you will know, let's for example, this SMA, its occurrence in India, like many other things, we do not know. And sometimes there are really frightening reports of number of carriers. And this is something where there is a problem in the splicing, and you get dysfunctional protein. So what we are trying to do is to set up a model. And as I said, there are some interventions possible, which are just prohibitively expensive. So we are trying to working with the company to make the drugs which are currently used, So can we make them at very low cost and try it in our system? We are making multi-gene cell-based assays or fly model for the disease and screen for hit molecules using the derivatives or the trying to repurpose the compounds or somehow manage the patent things and validate them within our labs. So in this process, we are now close to making the fly, which will have where we remove the fly gene with the human gene. So and then introduce the mutation, and because it's not, there is no new research here because this system has been shown to be working, where humanized SMN2 fly is made, and the flies actually show the disease phenotype, which can then be used for screening and finding the potential target. So we are in the almost finishing the phase 1 now. With phase 2 we should begin some activity. It's a very convenient system where large scale screening can be done and really costs nothing, the system as such. Then my colleague, most of the thing I'm going to talk about is done by Vasant Damodaran, who is the stem cell specialist in ticks. So here he is generating a Pompe disease model using the CRISPR-Cas9. So as you can see here, you have the gene, you can do the editing here, remove the G enzyme activity, which is shown here. And these are now you make pluripotent cells. You can see all the markers are there. And then go and check the sequence that you have precisely removed whatever was desired. And then you go and do the karyotyping and see that all the chromosomes are in shape, and you can actually create the disease like this control, and this is Pompe cells. So a Pompe human embryonic stem cell line lacking GA activity generated using gene editing, and the cell line maintained pluripotency and karyotypically normal. So we are more or less set with this. So we will do some more tests, and of course after that it should be good to go. Then there is this is now Pompe disease. Now we are making cardiomyocytes of this to recapitulate the disease phenotype. And this slide shows that both the control and GA mutant cells derived cardiac cells displayed cardiac markers in similar levels, and cardiac cells displaying disease pathogenesis previously reported in patient iPSC derived cardiac cells match to what we are seeing. So this encourages us that the technology, a good thing about all these technologies, once you have cell culture lab, then actually you don't need anything big in terms of facility or equipment. Sometimes some expensive chemicals are required for growing the cells, otherwise, which we are indigenizing. But otherwise, as Alok said, we want to really become Atmanirbhar, then only it is possible. Otherwise, we will be exploited by companies for a variety of reasons. Then we are also establishing the CRISPR-Cas9 based stem cell model for the very long acyl hydrogenase deficiency, which essentially shows here that we could do it successfully. Here is the gene locus. Here this shows that we have edited, because this extra band means there is editing is done. And when we grow the clones, then here there is a 22 nucleotide short fragment. And this shows that by sequencing that we have exactly done what is there in the disease, and the pluripotency is tested, all the OCT4, NANOG, and SOX2 are expressing. So this shows that we have a homozygous deletion mutant line, and the mutant express all pluripotency markers, and the further characterization of this thing, these are in progress. So similarly, we are setting up the stem cell model for DMD, and as you can see, I don't want to bore you, but it clearly shows that we have got this 183 based deletion, and there is a homozygous deletion mutant line, which we are now, several of them, which we are characterizing. So what we are also doing, so those are about the creating platforms where we can use various possible options or new interventions without really much of investment, and try to find the leads. So we want to do multiple things. So whatever succeeds, we can push that. So here is something called brittle bone disease, which we are working with the CMC Vellore. Uh, this in fact is quite an advanced stage where we get the fibroblasts derived from patients, which we create, we assess for the mutations that are there. So 10 most prevalent mutations are known. So we make iPSCs of those and then do the gene editing to fix the cells, and these iPSCs are then sent to CMC Vellore, where they can straight away put in their GMP facility and put in the patients. So this we are doing with Dr. Madhuri, who is very enthusiastic, and this program is quite. I mean, this is at the patient trial level, and is funded by DBT. So we are hoping that these are the things which will not only one disease, but we want very keen to set up a culture in which we can complement each other's expertise and accelerate the findings. So this is another you all must be knowing. We are also trying enzyme replacement therapy or protein therapeutics for alpha glycosidase, MPS6, alpha glycosidase, and then cerebrosidase. These are based on our clinician partners who suggest us what is doable, what is not doable in terms of patient availability and so on. So this is happening at different levels of progress. We are also making a database. One of our bioinformatician is now full-time involved in this. We call it Genetics, where we want to really add and make it 100% open to people, and develop a repository for clinical data, which should be provided by our collaborators. This is heavily dependent on our collaborators, and we are guided by them. And it is one point where I think we will have lots of interaction with Alok and his team. He has a real expertise in this first-hand expertise. So I think we will. So this now our front page is ready, and kind of architecture of this database is set. Now we are populating it, and we will soon try out with some users and then make changes so that it becomes as handy as possible. Now, in the next few minutes, I will talk about something which we are really excited, which is a mRNA-based therapeutic platform for genetic disorders. So this is, I may be giving some example, but really this is disease agnostic, because once the platform is set, then you can actually try out many things. So here the mRNA encoding a therapeutic protein is optimized. There are certain technicalities in that, and then lipid component, and then you make lipid nanoparticles, which can then be tried in model systems or a cell-based system, and hopefully it will go to patient. And by the tremendous success of mRNA vaccine, I think we are now very well positioned to use this technology, which can be really inexpensive for the therapeutic purposes. So this is to make an affordable mRNA-based therapeutic intervention to treat monogenic rare as well as prevalent genetic disorders in India. This is to replace the costly enzyme replacement therapy with an affordable mRNA therapy, and mRNA coding for biotherapeutic protein will be injected into the patient, and the functional therapeutic protein is produced inside the body with the identical most translation modification, etc. So lots of problems that are there with the protein therapeutics we don't have to face them. Cell will take care of that. So one of the things we are trying out here is the Gaucher disease, which and there is no particular reason, as I said, just because with our discussions or partners, something which appeared that it might go sooner. We put, but we can do multiple of these in simultaneously, because, as I said, it doesn't really require much resource. So we can put one scientist and or technician and all, he or she can follow up on disease. Reason for here is, as I was saying, that is very, very expensive disease. I mean, treatment is available, but it is terribly expensive. And we have really experts who have experience of this, and we are having industry partners here who are very generous to offer their facilities, like companies like Genova, who have given us an open offer to use their GMP and take any technology, and they really openly discuss with us. And there is reason why we are interested in this, because protein therapy, although it works, but most of it is lost, and we are thinking that the mRNA will be really very effective here. And similar design and pharmacokinetic parameters will work for Fabry disease and maybe some others. So here, this is the just simple outline of the things that you make the mRNA. I will not go into really technical details. Because I think this is not the platform for that. And make the nanoparticle try out in the cells and models which we are already making, and then try the animal model, and then go for human trials. So that is the strategy which is really applicable for whichever disease. And this is another one with exactly the same kind of thing. We don't need to do many things. Once we know how to make this RNA and how to make the lipid particle from this, and you have some way of testing in the plate, which also we have now the efficiency, and then models we can make, or we can test, or depend on how things stand for that particular disease, and go quickly. So
Dr. Mishra, so sorry to interrupt you, but if you could just
yeah, I have just one more slide and then we are done.
Okay, thank you.
Yeah. So there are now, I mean, this is very powerful technology, and this is our summary slide, that the large number of disease can be tried. We are having partners, and some we are improving our network, so that both research institutes, hospitals, and industry partners, so that when a solution is found, we can deliver it to the people. And just to impress you, as I said, that we are collaboration-based institutions. So we have, I'm listing out here only where we have a rare genetic disease effort associated activities either going on or on the plans. So as I said, in STEM, with CCMB, with NCVS, certain neurological disorders, CMC, I just gave you an example. This is a company that makes chemicals, drugs, which we want. Ashoka University, we just signed MOU, and we should begin soon working. The Bangalore Baptist Hospital, there's a RDI, MND Trust, Center for Human Genetics, and Indira Gandhi Institute, and JSS Hospital. So in fact, our team will be visiting JSS Hospital on 28th of this month. So we are very sincere and very really determined to take the technology that is there in our reach to make it possible to reach it to the patient, because for them the time is very important. And I think all of us have to work together. So particularly for TICS, which is a very young institute, so that we learn, we get guidance and help from all of you of various kinds of information, and we work together to impress the system, regulatory authorities, so that these benefits of science reach people as soon as possible. So that was just brief summary what I wanted to say. So thank you very much again for the time that you all gave to me, and we can discuss. As I said, I'm not expert, but but we can discuss, and I can answer some questions.
Right. So also, if Dr. Bhattacharya, you can switch on your video, then we can just take a quick picture of the speakers for this session. I'd like to thank you, Dr. Mishra, for such an excellent overview, and particularly excited to see all the work that TICS is doing on rare genetic disorders and the tremendous potential for therapeutics that lies in that work. I think as a patient, that's very exciting. So we have a question from Dr. Sudha Bhattacharya. You can ask your question.
Yeah, Rakesh, really very impressive talk and very encouraging for us because, as you say, once the platforms are in place, then one can use it for a lot of other diseases. But so my question, very briefly, one of the things that you talked about in the osteoporosis imperfecta, that you are actually gene correcting using CRISPR
Yeah.
and then generating cell lines which are corrected, and then they can be given back to the patients. So my question is that, are you collaborating with some industry to scale it up? Because I think the bottleneck there is that the number of cells, it may not be large enough for a pharmaceutical purpose. So what are your plans in that?
So, so we don't have, I mean, the whole key here is really a GMP facility, which CMC has, we don't have, so we cannot make something which can go to patients. So, and CMC has, so that's what our, I mean, this has begun just maybe about 6 months back. But you raise very important point for, even for the gene therapy. Recently, I was talking to somebody in a short visit to UCSD where they're working on some genetic disorder, gene therapy. And they say that most important part is really GMP facility where you come in large amount because they don't want to cells. So the take the patient cell and do large scale transformation and and put back the cells in the person. So GMP facility are the crucial things. That is something we have to work with the hospitals so that it's three-way thing. That industry who will amplify and should be in really close contact with the clinicians who are really going to use that. So the short answer is that we haven't identified industry partner, but at the moment, since this is a trial level is going, but you are very right. I think we have to now work together to set up few, it's not set up, put in a few GMP, as you can see that many of these things we are all having in the lab. Soon we will need these kinds of things.
So we have a question from Dr. Alok Bhattacharya. If you can keep it very brief because the next session will start soon.
Yeah, I just added to what Dr. Rakesh Mishra said, that in fact, TIGS and us are organizing a brainstorming meeting on messenger RNA therapy, the various aspects of that. That's coming up next month.
Right.
And all the people in the country involved in it. And this is a joint effort with TIGS. This is just to develop. I think I personally also believe that messenger RNA, like he was very enthusiastic, I also feel that messenger RNA therapy has a probably much easier to deliver compared to all others.
Yeah, so we're excited to hear about that collaboration on messenger RNA and potential therapeutic applications coming from that. Thank you so much, Dr. Mishra, Dr. Bhattacharya, and to all the people who are listening for this very exciting and